Kiravana?
Dr. Ravindran, can you hear us? Dr. Ravindra, can you hear us?
Hello. 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 అంటే మా ఫోన్లో కరెక్ట్ వచ్చింది లక్ష్మణ మా కరెక్ట్ వస్తుందా ఒక్కసారి బయట వెళ్తా మేడం యూట్యూబ్లో డీబీటీ సిడిఎఫ్డి డీబీటీ సిడిఎఫ్డి Okay, madam.
Namaste, madam.
Hello. Good afternoon, ma'am.
Hello, good afternoon.
How are you, ma'am?
Good, good. Good that you are arranged. Nice.
Yes, ma'am. Shortly we will be starting. Just wanted to know whether whether the voice is audible and
Yeah,
in.
voice is audible. Uh, uh, Madam Pramila ji, Mrs. Agrawal has joined.
Hello,
So,
ma'am. Good afternoon.
Madam, good afternoon, Dr. Gambhir here from Pune.
namaskar, namaskar, Dr.
नमस्कार.
Gambhir. Good to see you.
पुण्यात आहात?
हो, मी पुण्यात आहे.
अरे वा वा.
Dr. S.K. Mishra, Professor S.K. Mishra of SGPJ Lucknow also has joined.
Right, right, madam.
नमस्ते मैम डॉक्टर रविंद्र
Ravindra. Oh, Ravindra is also joined. Nice to see you, Dr. Ravindra. Yeah, it's a very nice way to meet everybody and so easily. I can see Nirja. Hello, Nirja.
नहीं जरूरी नहीं।
All these introductions what required I was giving for them. Okay. Hello, Shubha ma'am, can you hear me?
Hello. You are audible?
Yeah. Thank you, ma'am. Actually, I started with CDFD introduction, but I was giving to only our people, so I'll give a repeat
Oh,
what I have
okay.
told till now.
We thought you hadn't started.
Yeah, no, ma'am. So.
Okay.
So I was just telling that, I welcoming all online participants, media persons, and CDFD staff and students over here. And then today I welcome you all to this symposium on genomics initiatives of by Government of India, titled Genomics of People, by the People, and for the People. So want to tell something about CDFD to the people who are online. It is a premier institute which is funded by the Government of India, and CDFD is renowned for its pioneering work in fingerprinting, DNA fingerprinting, as well as genetic diagnostics, and also in cutting edge genomic research. So we offer services for rare genetic disorders as well as inherited disorders, and forensic investigation, and also plant fingerprinting testing is done over here. So with a strong focus on translational research, training, and public health initiative, CDFD plays a very vital role in, advancing precision medicine and genomic science in India. So, with the advent of the next generation sequencing, which played a very important role, and it is continuing across the fields like agriculture, forensics, science, and also in healthcare. So, especially in human genetics, so the novel or a novel gene variant identification, it is really helping us in leading to better management as well as development therapies also. Thus, it is addressing our global challenges on healthcare system. So today's symposium is a part of annual memorial lecture series, which is conducted every year in memory of Dr. S.S. Agarwal, who is considered as a pioneer in the medical genetics education in India. So now I request the online participant to mute yourself, please. Yeah. Thank you. So now I request Dr. Shubha Phadke to introduce the gathering about the contributions of Dr. S.S. Agarwal. And as Dr. Shubha Phadke ma'am is a former HOD and in the Department of Medical Genetics in SGPGI Lucknow. And one important thing is she's the first DM student in medical genetics with Dr. S.S. Agarwal. So over to Shubha ma'am.
Thank you, Dr. Usha, uh, for the introduction and for organizing this program. Dr. Ashwin, Director CDFD, and our Madam Dr. Pramilaji Agrawal in the who has joined online. Dr. Madhulika, Dr. Nirja, and many friends I see online for joining this program. I welcome them all and thank them for joining us. I also see respected Professor S.K. Mishra, sir, from who was the one to start telemedicine facilities, probably pioneer in India and first time in SGPGI Lucknow. I'm happy that he had joined here, and I'm happy that because of such telecommunication facilities, I can be a part of such a important gathering without traveling. Though I really wanted to be to come and join for the program in person, I could not. But I'm really happy that I could be part of this program because it's very close to my heart. In fact, this is a very emotional program rather than scientific or medical symposium because it is in the memory of our sir, Dr. S.S. Agrawal sir, we usually use say SSA sir. So in his memory, we are holding this program every July of July, and basically the theme of the program is kept in the form of sort of public lecture. And at the same time, it helps all of us to pay him respect, to remind ourselves what we had to do further and how his contribution has shaped us. His contribution to medical genetics in India is immense, and all of us who are ourselves called as medical geneticists really owe our existence or our identity as a medical geneticist to him, because he started the DM medical genetics program first time in India in HGPGI, and he started the department and then he started this program. And now what we can see, at that time, nobody knew what genetics is. As I said, nobody thought that telemedicine department has some utility. Same way, even medical genetics was considered. Everybody thought, they felt that it is something in research and not really for patients. But now we know that medical genetics has taken a center stage of practice of medicine. So anybody who has to practice medicine has to know genomic vocabulary, and probably many of them are ordering genomic testing as well, as the next generation sequencing has become probably the first test for any patient who is supposed to or thought to have a genetic disorder. So at this point of time, there are so many medical geneticists in India to take up the challenge of practice of medical genetics, as well as to teach medical genetics to students, and not only to those who want to become a medical geneticist, but it is essential that every clinician who wants to practice in the era of molecular medicine needs to know medical genetics. And that's why what we are doing, and many of our students are who have passed from medical genetics department in HGPGI and AIMS and PGI Chandigarh, then Gangaram Hospital. So many of them are doing the same thing, trying to spread awareness as well as educate clinicians in medical genetics. On this background, the other important thing which has come up is that these next generation sequencing techniques, they are useful for diagnosis of genetic disorders, but they are also useful to look at the genetic predisposition of each and every disease. Practically accident, leave apart accident or so vitamin deficiency, most of the disorders have some genetic predisposition. And that is why a lot of research is going on, the understanding the genetic predisposition to, say, schizophrenia, ischemic heart disease, ulcerative colitis, age-related macular degeneration. So name any disease, and you have research papers on that, understanding, trying to understand genetics aspects of that disorder. And for that, so many people are undergoing this exome sequencing, either for themselves or as a part of the diagnosis of their some family member, or as I said, as a part of research protocol. So when so many now so-called normal people or lay persons are undergoing this NGS testing or genomic sequencing testing, its time has come that not only clinicians but lay persons need to know about their genome, as well as what these whole genome sequencing can do to the patients, persons, and everybody. So what I mean is that its time has come that we have improved genomic literacy among the lay persons as well. So the program, today's program, what we had already doing, that this lecture preferably is targeted towards creating awareness amongst lay persons as well. And that's why we want to keep it simple so that those who are not medical people, those who are not geneticists, can understand the beauty of genetic research, what is going on, and as well as its applications in clinical practice. So I really thank Dr. Ashwin for organizing this program and selecting a very interesting topics which are latest as well as for the whole population, and I'm sure all of you will enjoy this information. So this information for laypersons is also important because like any other medical specialty, genetics also has lots of ethical dilemmas posed in front of the patients and clinicians. And probably genetic research has created lot of ethical dilemmas in front of everybody. And that is why Human Genome Projects had a separate cell for research into the ethics and legal issues caused by human genome sequencing. So, as these dilemmas are coming in front of us, say identification of unrelated genetic variations in the individual for late onset disorders, for cancers, for sudden death, then variations of unknown significance. The idea proposed that we should sequence everybody to find out whether they can have a child with a serious genetic disorder or not. So all these issues need opinion of the society, and that opinion of the society should reflect not only of the clinicians or medical geneticists, but the lay persons, the leaders of the society, the educationalists, the teachers, the social workers, sociology people, religious people. So it is important that all of these people know this cutting edge research in the field of genomic medicine. So I really again thank Dr. Ashwin, Dr. Pradnya, Dr. Shagun, and their team for organizing this program. And it's probably even this, as I say, it's really my personal thank you. So it is not only from the society or Department of Medical Genetics, but I know that how important this day is for myself and also for my students. So, with these words, I would really again thank everybody. And you will see that this program, how interestingly it has been planned, including what I liked very much is the essay competition for the children. So, this is something new which Dr. Ashwin has added. And I think this makes this program very, very special. I again thank Pramila ji and all others who have joined online. I think there must be all CDFD staff there, and I wish I could be with you and had opportunity to meet you all. Thank you very much. Thank you, Usha.
Yeah. Thank you, ma'am. That was really great. And we also wish that you were here. And it was so nice, like how you told that next generation sequencing and especially exome sequencing, genome sequencing is going to help to identify the genetic disorders and how to benefit the society. Thank you very much for that. So with this, we go to today's program. So today's program is basically about genomics initiatives of Government of India. So we are going to highlight about 4 different programs, government programs which are running. And the first program is Genome India project, about Genome India project. So this Genome India project was recently dedicated to the nation by our Prime Minister, Indian Prime Minister Modi ji, where 10,000 genomes are sequenced. So Dr. Divya Tej, a scientist from CCMB, who is involved in this project, is going to deliver and tell us how they have progressed and about the project. Tej.
I don't know. नहीं नहीं वीडियो यहाँ लगाओ ना। हाँ नहीं नहीं यहाँ फेसिंग यहाँ। उसको पिन करो ना। ऐड पिन हाँ सेकंड हाँ आया क्या हाँ ओके
Okay, good evening. Am I audible? Yeah. So first of all, I commend Dr. Ashwin and the organizing team for putting up such a wonderful program together. And also, thank you for inviting me here. It's an honor to talk as part of this series. My name is Tej. I'm from CCMB, and we've already heard the importance of understanding the genetic basis for disorders. And Shubha ma'am has actually given a very nice introduction about why it's important to study this. And today I'm going to talk to you about a project, a flagship project called the Genome India project, which is launched by the government of India to sort of tackle this from an Indian perspective. So almost all sorts of diseases are controlled, or there is a genetic predisposition for almost all sorts of diseases, right? And when people started using the existing data, which is typically Eurocentric in nature, they realized that the same thing cannot apply everywhere. And even more so in case of India, because we are not one kind of people. We are a heterogeneous set of people. We have thousands of languages. Am I audible to the online audience?
Yes, yes.
Yeah, okay. Yes. We speak many different languages, which can be sort of broadly classified into 4 linguistic groups. And we have diverse biogeographies. We have the Himalayan range, we have deserts, we have Eastern Ghats, Western Ghats, and whatnot, and all the adaptations that come with it. And more importantly, we are segregated into these tightly knit communities who have very different socio-cultural or ethno-linguistic attributes. So very quickly, I'll run past this slide, which is a previous work from CCMB, which has shown that there exists a lot of private genetic diversity. I mean, without going into the technical details, what this means is that there is so much of genetic diversity that happens within a community that is not captured in a typical sampling. Because unless you go and specifically sample those communities, you will not capture that. So keeping this in mind, we of course want to capture as much genetic diversity from India as possible. But when you look at the existing data, this is from 2022. So we contribute. Okay, fine. I think that's fine. Works. Right. So we are the most populous nation in the world, but still, if you look at the contribution of our samples to the global genomic data set, it's less than 2%. And which means that most of the existing data set will not be applicable to us. Keeping this in mind, the government of India started this project in early 2020 with this grand idea that we will sequence 10,000 Indians. And these are not just randomly sampled. These are sampled spanning the breadth and length of the country, cover 83 different communities. We have actually collected 20,000 samples out of, and all these are actually extensively phenotypically characterized. And I'll cover what type of phenotype we have covered. And the idea is that we create this catalog of genetic diversity for Indian populations. And we did that using whole genome sequencing. And this was a massive feat that actually saw that almost 20 different institutes across the country have come together. And these are the participating institutes. So from Hyderabad, both CCMB and CDFD are part of it. So the project is spearheaded by CVR at Bengaluru. And these are the remaining institutes. So there are several different sample collection centers, then there are method development centers, and there are 4 main sequencing centers which have shared the workload of sequencing these 10,000 samples. And each marker that you see on the map is one of those 83 communities that I spoke about. And their color indicates what linguistic group they belong. So again, the map is also shaded according to the linguistic group that is predominantly spoken in those areas. So you have the Indo-European group, which is mostly at the north and central India. Then you have the Dravidian group of languages, which are spoken in southern India. And then you have the Tibeto-Burman set of languages, which span the Himalayan, like very North India to Northeast. And you have these pockets of small languages, set of languages called Austroasiatic languages, which are predominantly spoken in some tribes. And this is the overall workflow of the project. So the idea is we heard how important it is to make sure that the ethics are done right. So we have contacted about 24,000 people, and we have taken informed consent from all of these. And these consent forms were available in 23 different local languages, so that they could really, it's not just a signature on the form, but they could understand what they were saying yes for. And of course, a lot of people said no, and therefore they are not part of the study anymore. And out of that, 20,000 odd have said yes. And from them, we have done a bunch of phenotypic analysis, which includes anthropometry, which is their height, weight, and waist circumference, head circumference, and so on. Then we have collected blood samples. Part of that blood has gone for DNA isolation, which was needed for further genetic analysis. Part of that blood was also used for blood profiling. Again, we've covered a bunch of parameters, which I'll show in the next slide. And an aliquot of all these samples was also biobanked at CBR for future research, right? And then we do a QC, initial QC using array-based genotyping. Again, for the purpose of this talk, I'll not cover it, but essentially it's a smaller version of understanding what the genome is of that sample. So that once you okay it at that QC level, that is when you take it to a whole genome sequencing, which is way more expensive, right? And then we do the data analysis of these samples. And the data is now available at one central point, which is IBDC, or the Indian Biological Data Center, which is housed in RCB at Faridabad. So sampling, this was some pictures which actually as soon as we started the project, COVID had hit. The project was sanctioned in Jan 2020, then COVID happened. So there was a lot of challenge in going and collecting samples. In fact, you can see that once things opened up, we had to follow the social distancing, etc. And these are all in remote places. It's not in any urban setting, right? But we still did that, and so sampling took quite some time. Then, like I said, these are the various phenotypic data that we collected. Anthropometry, which includes the typical age, gender, and other physical attributes. Then we do the common hematological parameters like your hemoglobin content or CBP, etc., etc. And then we did some kidney function test, liver function test, and as well as glucose-related tests. All this data is available for most of the samples. The idea is that once we generate all this catalog of genetic diversity, we try and ask what those genetic variants tell you in terms of the phenotypic outcomes. Like if there is any population, let's say, that is predisposed to diabetes or obesity or just heart disease, etc. Essentially, because most of the workload was done by 4 different centers, we had done a rigorous set of QC protocols. So again, I will not go into the technical details, but the idea is that we had reference samples which were sequenced at all the institutes, and then we compared the results. Only when we knew that those the results for the reference samples have agreed, that's when we were okay saying that, okay, even if the work is distributed at 4 different centers, we are getting the same results. And this kind of data is a very large data set. Okay, every human genome sample that you sequence ends up creating something like 50 to 60 GB of raw storage or raw requirement. And after that, as you process it, you end up generating something like 150 to 200 GB of data. And that multiplied by 10,000 means that we needed over 2 petabytes of storage. That's like, I don't know, a million songs And doing this process also was very computationally intensive. We had to distribute it and do it. And it was an upward of 0.7 million CPU hours. So which is distributed on high performance computing systems, which come with thousands of cores, right? So, and the reason I'm pointing this out is because it couldn't have been done by one individual. This was a computational feat that was sort of the first in the country, and it could only be achieved because a lot of premier research institutes have come together to take up this challenge and contributed in their own way to make sure that we could achieve this task. And the final output file is, like I said, is now stored at one place at IBDC. So what do we know from this? I will very briefly cover what some basic preliminary findings that we have so far from the project. So first thing, we have, so after QC and everything, we have data for about 9,700 individuals. And we identified more than 130 million variants. And now this is a large number for a project of this size. It's a very large number. And our preliminary analysis tells that most of these variants are actually unique to Indians. They are not covered in other data sets, which is expected. And most of them are also rare and ultra rare, which means that they're present in very small proportions in the population. That's because they're unique to one particular community or ethnic group, which is why unless you specifically sample them, you will not pick that. So it's not present in other ethnic. So we captured a lot of that kind of private genetic variation. And when we overlap this with a database called ClinVar database, which has a high quality collection of clinically relevant variants, what was surprising or interesting is that about 58% of these individuals had at least one ClinVar variant present in them. They could be carriers for this, and etc., which means that they're apparently healthy people. Of course, if they're carriers and heterozygous for this, we won't expect a phenotype. But this is a finding that has its own merit, right? And this is just the beginning of what we can uncover with this kind of a data set. Now we start overlapping this data with phenotype. We can get a lot more information of how genetics dictates all these traits. And the reason why I showed about the linguistic groups and the map is because I wanted to highlight how language and geography are proxies for genetic diversity. So this map I've shown you earlier. The plot you see on the right is called a principal component analysis plot, or a PCA plot. Essentially, what it tells you is it's a compressed view of your entire genetic similarity. If two dots are nearby, then they are genetically similar, and if they are far apart, then they are more different, right? Which means that if you see a bunch of dots together, they are genetically much more closer together. And what you can see is this neat climb. In fact, this is reported in previous findings. So this is called the North-South Indian climb. So you could see all the people who are collected from the Indo-European part are sort of on the north part. And slowly, as you come towards, all this Dravidian groups start coming up. And then you have neat clusters of Tibeto-Burman and Austroasiatic tribes forming together. And what I've not shown you is a PCA colored for biogeography. And there also we pick clusters that those which are separated by, let's say, different biogeography also cluster together, which means that both language and geography can be used as proxies for genetic diversity. And one last preliminary finding. So this is called as, this is a preliminary analysis on what is known as pharmacogenomics. So typically when you go to a clinic, you're prescribed the average dose for everyone, right? Like you go with fever, you're prescribed 500 mg of paracetamol. But now we know that people metabolize drugs in different rates, and that is controlled by what kind of metabolizing alleles you have. Some might need half the dosage, some might need double the dosage, some might need a different drug altogether. So the branch of genomics which deals with understanding this is known as pharmacogenomics. And what we have shown is some pharmacogenomic alleles that we have identified from Genome India. But I want to highlight just one thing. This is this particular variant, which is DPYD, and it is present in low frequencies in Genome India, But the reason why it's important is that it has a direct effect on how you metabolize these two drugs which are used for cancer treatment, which means that if someone from these populations who have these variants are prescribed this, it may not work the same way that you expect. And again, this is just barely scratching the surface of what is possible with this kind of data set. And we are currently in the process of analyzing this data set. So this is sort of a summary slide that I have. So what we have achieved and what we hope to achieve. So we have done inclusive sampling by deliberately oversampling underrepresented communities. Then we have biobanked these samples for future research, and all these have detailed phenotyping. And this project was an effort in itself and built a lot of capacity that we can leverage for future such endeavors. And what we hope to get is what kind of ancestral or migratory patterns that we have in our populations, what kind of disease risk we can predict. We can build a array, which is array is basically, like I said, a smaller version of analyzing your genome, which is much more cheaper. We can build one array which is probably much more geared towards Indian genotypes compared to what we use normally. It's a step towards precision medicine, which tackles with describing or prescribing medicine specifically to that individual based on their genetic makeup. Imputational panel is something used with the array, which I will not go into. It has seen a lot of novel computational method development. And these are digital public goods that are available for others to use in their own research. And hopefully this will pioneer some translational research in the country. With that, I'll stop. Like Dr. Usha mentioned, this was dedicated to the nation by the Honorable Minister of Science and Technology and our Prime Minister, and I'll be happy to take any questions. Okay, sorry.
Thank you.
Questions later, Jason.
Thank you, Tej, for such a clear overview of the Genome India project. And I request all the participants as well as the online participants also to put their questions in the chat box so that we can discuss in our interactive session. So with this, we go to our second project. So the second program, ready? Yeah, the second program is Mission Program in Pediatric Rare Genetic Disorders, which is funded by Department of Biotechnology. And this is a pan-India project involving 16 centers, out of which CDFD is the nodal centers, so nodal center, and which is held by Dr. Ashwin Dalal, and he is going to talk about this project.
So very good afternoon to all of you, and we'll So we have just now heard from Tej about the important program that is the Genome India project, so which was a project to generate data. And what I am going to talk to you today is a project which is working towards utilizing this data, whatever these kind of projects like the Genome India or other projects generate, how we utilize this for patient benefit, or how we utilize this to identify some new things towards diagnostics or towards therapies for patients with different genetic disorders. So keeping in mind the importance of the rare genetic disorders, the Department of Biotechnology 2 years back embarked on this program called as the Mission Program in Pediatric Rare Genetic Disorders, with the objectives mainly of recruiting patients, doing their genetic analysis, and working towards affordable diagnostics and some therapies, as well as awareness programs. And the main USP or the main importance of this program lies basically in the joint activity of both the clinical centers across the country as well as the basic scientists across the country who are jointly working towards identifying the causes of these rare diseases as well as finding the mechanisms or how these diseases lead to these different conditions. Okay. So the main activities under this program are basically recruitment of patients, where we recruit patients from different centers, undertake their whole exome, whole genome sequencing, that is sequencing the all the genes in the particular cells. Where we find a cause for disease, we do counseling for those families. We explain to them what is the disease and what can be done. Whatever data is generated is kept in a resource portal where all the database of variants is kept. We analyze the variants using different in silico, that means computer-based methods, as well as using different cell line or Drosophila, zebrafish, or mouse models. These models help us to understand these diseases better. And then we also have a program working on the gene therapy for some of these disorders and the awareness programs about these diseases. The first important step is to collect data, and this collection of data is very important through a standard operating procedure so that the data is collected in the same way across all the 16 centers. And that is why we have an elaborate online collection of data where all the details of the pedigree of the patients, the patient details, their height, weight, their clinical features, all this is collected in a systematic way so that it is useful to analyze it in future also. Even when this project is over, all this data will be available for future scientists to make use of it. And it follows a set pipeline where there is a collection of data, sequencing, data analysis, variant identification, pathogenicity prediction, then functional analysis using different model organisms, and genetic counseling. And this systematic protocol helps us to maintain this protocol across all the samples that are received. So as of now, the aim is to do this analysis in about 5,000 patients over 5 years. And till now, almost about 1,800 patients have been registered. We have completed doing exome sequencing for about 1,000, genome sequencing for some of them. And of these 1,000, about quarter of them, that is about 257, we could find a variant where we could explain to the family what is the cause of disease in that particular family, as well as we could find some candidate new genes which are undergoing further characterization. The second objective was basically to develop a database, and that happens through an elaborate website that is available where all the information about work done under this project is available. As well as we are working, and soon this database will be available, called Indwar, where we want to collect all the variants in the Indian patients and link it to the basic scientists who would like to work on those particular variants. And thirdly, all this data is stored in IBDC, which is the repository of data in New Delhi, where all this exome and genome sequencing data is stored so that it can be used in future by other scientists also. We also have establishment of the lymphoblastoid cell lines. So these are cell lines which become immortal. So they do not die. These cells keep on dividing. And that becomes a perpetual supply of these live cells which can be used for different functional assays in future. So for a number of patients, we have been able to transform them into these lymphoblastoid cell lines. Then regarding the functional analysis, we already identified a novel gene called as serpin A11 as a novel serpinopathy. And this is being characterized now in the mouse model. So this was one of the achievements of this project, to identify a novel gene. Another gene we are still working, the group here on Drosophila led by Dr. Rohit Joshi, they are working on AIMP2 as another novel gene using a Drosophila model. Similarly, we use different assays, like here the yeast-based assay was used to classify a CDK5 variant in a patient with intellectual disability, or we use the splicing assays, and multiple different types of functional assays are used to reclassify these variants which are identified in patients with different diseases. We also have a group working on development of panel-based diagnostics for mitochondrial as well as other panels. And we are also working towards a new technology, that is the long read sequencing, which helps to identify the structural variants which are not able to be identified by the routine exome. So it's a joint project working on different aspects of human genetics, trying different ways to identify the genetic cause in almost all patients with genetic diseases. We also have a collaborator in CMC Vellore who is helping us to work towards the development of therapy for some of these disorders. One group they are working on is the Fanconi anemia. It's an anemia that is aplastic anemia, where the production of blood cells stops from the bone marrow. And here they have been able to do the transduction of these lentiviral vectors. So these are gene therapy vectors which are made, and they have put them into the patient fibroblasts, and they have shown that the patient fibroblasts now start functioning in a normal way after transduction of this lentiviral vector. Similarly, they are also working on another disease called as congenital dyserythropoietic anemia, a type of anemia where again they have used the lentiviral vector to put it into the cells, and they have shown a significant reduction in the disease. So in future, they plan to use the mouse model to show this same effect, and then the next step would be to go into the humans. Similarly, we have a lot of awareness program across the different centers across the country, where awareness programs are done in medical colleges, in schools, about these rare diseases, because more and more awareness will help us to get more and more patients who can benefit from these diagnostics. We also have a lot of social networking presence in YouTube, in Twitter, LinkedIn. This is mainly again to increase the awareness about the rare diseases, and especially because of the newer treatments coming up, it is very important that general public is aware of these rare diseases. We have achieved a number of publications through different scientific as well as in the common press to increase the awareness about these disorders. And that's the advanced laboratory that has been established along with this major instrument called as the NovaSeq X Plus, which reduces drastically the cost of doing genome sequencing. The first human genome was sequenced with $2 billion. Up to last year, we needed about 1 lakh or more rupees to sequence a human genome. With this new instrument, now we are able to sequence it in less than 30,000 rupees. So that's the drastic decrease in the cost, and that helps us to do more and more of this analysis to help identify patients with these genetic disorders. So that's the great team from all the centers, and that's the team working in CDFD here towards achieving these goals of the mission program. We still have about 3 years left, and we hope that we are able to recruit all these 5,000 patients and try and identify more and more genes responsible for these different disorders. So thank you, and we'll take our questions, if any, in the last session. Kushan? PPT. There. Mm.
So the third government initiative project is DBT flagship program, that is UMMEED, which stands for Unique Methods of Management of Inherited Disorders. So basically it is for establishing the genetic labs in medical colleges as well as district hospitals. So now I request Dr. Pregna, who is a professor in the Department of Medical Genetics, NIMS, and she is also a part of the Nidan Kendra and Training Center under UMMEED programs, to just tell about the project.
Thank you, Dr. Usha. At the outset, I'd like to thank Dr. Ashwin for this opportunity. And before I begin, I would like to pay homage to Professor S.S. Agarwal. Sir was indeed a visionary, and he is instrumental in starting medical genetics education in India. And of course, if it hadn't been for Sir, most of us medical geneticists wouldn't be here today. So with that, I'd like to move on to the topic. So I'll be talking about the DBT UMMEED program. UMMEED actually is an acronym. It's a clever play on words. UMMEED, we all know, is the Hindi word for hope. But UMMEED actually is also an acronym which stands for Unique Methods of Management of Inherited Disorders. So this is a program which has been initiated by the Department of Biotechnology, Government of India. And why do we need this program? So we just heard a series of lectures on all the important advances related to genomics that are going on in India at present. And if we have to ensure that these advances reach the grassroots, really benefit the population, really reach the patients suffering with these genetic disorders, we need to have a program which reaches all the way up to the very primary care level. So the entire program has been designed keeping this in mind, basically making genomic genetic technologies accessible to even the poorest sections of society. So the burden of rare diseases in India, if you look at it, there are about 7,000 known rare diseases. And worldwide, the estimated number of patients suffering with rare diseases is about 350 million. In India alone, the approximate number, of course, this is an estimate, nobody knows the real numbers, would be somewhere in the range of around 70 million. So that's a huge number. And although we call these rare diseases, individually they may be rare, but of course, collectively, they account for quite a big burden. And usually, because they are rare, a lot of doctors even are not aware about them. And typically, it may take an average of up to 7 years to diagnose such patients. 30% of patients with rare diseases die before the age of 5 years. So again, a lot of morbidity and mortality associated with these conditions. So keeping all this in mind, the Department of Biotechnology came up with this UMMEED program, and the whole initiative is designed on the concept of prevention is better than cure, because we know that only about 5% of genetic disorders actually have some sort of treatment available. The rest of them are primarily dealt with symptomatic supportive care. So obviously it makes more sense to prevent the occurrence of these conditions by early detection and early prenatal diagnosis and other methods. So genetic disorders account for about 80% of the rare disorders. So the mandate of the DBT UMMEED program is to contribute to the infrastructure and human resource needs for management of genetic disorders and to facilitate the use of cutting-edge scientific technology and molecular medicine for extending this universal health coverage for all. So UMMEED is also a significant contributor to the implementation of the National Policy for Rare Disorders, which has been initiated or launched by the Government of India in 2021. And I'll also come to that in one of my later slides. So what are the main objectives of the UMMEED program? One is establishment of genetic diagnostic centers in different parts of the country to provide patient care services. Two, to create manpower to deal with genetic disorders, and mainly to train medical personnel in clinical genetics and genetic diagnostics, because sadly, although we are talking about all these advances now in India, the medical curriculum, the MBBS curriculum is still pretty lacking in medical genetics. Very little is taught to MBBS students. So a lot of doctors actually graduate without having much knowledge about genetics. So it's extremely important that the knowledge of medical genetics reaches every doctor so that they can be aware of all these conditions and make the appropriate diagnosis in the patients and thereby offer them appropriate management. Also, to create awareness about genetic disorders amongst clinicians and the general public, spread the reach of diagnostic facilities for genetic disorders, prenatal diagnosis, and population-based screening for prevention, and also contribute data for future research in genetic disorders. So the Umeed approach is actually a three-pronged approach, as you can see. So on the one hand, you have these Nidan Kendras. On the other hand, you have these training centers. And at the third side of this triangle, you have the aspirational districts. So what are those? So basically, the Nidan Kendras are nothing but genetic diagnostic units which are being set up in government hospitals and medical colleges. Training centers are being established to provide training in medical genetics to faculty from government hospitals so that they can go back to their respective centers and set up genetic centers at in their own hospitals. And outreach centers are being established in aspirational districts for antenatal screening for prevention of common genetic disorders and birth defects, and for newborn screening for early detection and management of common treatable metabolic disorders. So Nidan actually is again an acronym, again a clever play on words. It stands for National Inherited Diseases Administration Kendras, and also Nidan is the Hindi and Sanskrit word for diagnosis. So again, Nidan Kendras are nothing but genetic diagnostic units which are being set up in the government medical colleges, government hospitals, and also zero-recognized not-for-profit medical colleges and hospitals. So funding support is provided by DBT to procure equipment and to support the manpower and consumables for the first 3 to 4 years of the establishment of the center. And thereafter, the host institute is expected to take over the running of the lab. The training centers are actually medical colleges, hospitals, research institutes, academic institutes, which have a minimum 10 years experience in research and training related to clinical genetics. So these are the centers who are eligible to apply for this training center component. And they are supposed to provide training in genetic diagnostics, which includes biochemical, cytogenetic, and molecular genetics related testing and clinical genetics. So only permanent faculty from government medical colleges and institutes are eligible to apply, because the whole idea is that they have to get trained and then go back to their respective centers and set up the Nidan Kendras and the genetic diagnostic units in their own hospitals. And of course, as a part of this program, it has been mandated that each of the training centers should train at least 12 faculty in 3 years, that is at least 4 per year. Moving on to the outreach centers in aspirational districts. So these actually are supposed to be supervised or taken care of by centers who have requisite infrastructure and expertise to carry out genetic screening of pregnant mothers and newborns. And each of these centers can adopt one or more of these aspirational districts. So these screening centers are set up in the district hospitals of these aspirational districts. And the activities that they're supposed to be conducting include screening of antenatal mothers for common genetic disorders and birth defects, newborn screening for some of the common treatable metabolic disorders, which, if detected early before the symptoms set in, can actually be very well managed with very little morbidity and mortality. In fact, they can be near normal if they are detected early enough, and if the treatment is started early enough. So some of these easily treatable common metabolic disorders include congenital hypothyroidism, congenital adrenal hyperplasia, galactosemia, biotinidase deficiency, G6PD deficiency, and some others. Then genetic counseling of couples with high risk of genetic disorders in their offspring are also identified in these aspirational districts and appropriately counseled so that they can go for timely carrier testing and prenatal testing to prevent the birth of children with debilitating disorders. And training is provided to paramedical health workers to enable them to identify such families who need such help and counseling. So the first phase, the pilot phase, was actually launched in 2019. But before that, there was this call for proposals, which started in 2017. It was a competitive call, and there were lots of applications from across the country who applied for all these three components: the Nidan Kendra component, the training center component, and the aspirational district component. And 3 rounds of shortlisting happened. And finally, 5 Nidan Kendras were chosen. So one of them was our center, that is Nizam's Institute of Medical Sciences, Hyderabad. So the program was officially launched in September 2019 in New Delhi by the then Honorable Union Minister for Science and Technology, Dr. Harshavardhan. And the first phase, as I mentioned, there were 5 Nidan Kendras and there were 6 training centers. So the Nidan Kendras were LHMC New Delhi, AHRR New Delhi, then our center, NIMS Hyderabad, NRS Medical College Kolkata, and AIMS Jodhpur. And the 6 training centers in the first phase were AIMS Delhi, SGPGI Lucknow, CMC Vellore, MAMC Delhi, CDFD Hyderabad, and NIIH Mumbai. So more than 60 faculty from government medical colleges and hospitals across India got trained in the first phase. And many of these trained faculty have now gone on to, they have applied for the Nidan Kendra to be established in their respective parent institutes. And many of them have been granted the Nidan Kendra project in the second phase of the UMEED program. The outreach centers, actually, in the first phase, there were 9 aspirational districts identified from 6 states, which were being mentored and taken care of by some of these apex institutes, which I listed before. And during this phase, a lot of patients benefited. So antenatal screening was offered for close to 95,000 pregnant mothers and newborn screening for more than 60,000 neonates. And this helped to prevent 34 births of hemoglobinopathy affected children, which included both beta thalassemia and sickle cell anemia. And also many couples were identified who were carriers who had the potential risk of having offspring affected with various genetic disorders, and they were counseled about going for appropriate prenatal testing in their subsequent pregnancies. And more than 350 newborns received timely diagnosis of congenital metabolic disorders, based on which early initiation of therapy was possible. So now that was the first phase, and now this is the further expansion plan. So the second phase has already been initiated and is going on right now, and a third phase is planned as well. So in the second phase now, there are many more Nidan Kendras, many more aspirational districts, and many more training centers. So if you can see the colors, to begin with, there were only a few states which were covered, the Nidan Kendras and the aspirational districts. And now you can see in the second phase, most of the country is covered, most of the states are covered. And in the third phase, the idea is to cover the whole, states from across India, all states, so that even the remotest corners of the country, the remotest corners of the nation benefit from this program. So this is the list of Nidan Kendras in the second phase. And you can see now all the multiple dots over here, which represent the Nidan Kendras, the aspirational districts, and the training centers under the second phase of the program. So at CDFT, what were the activities under the DBT UMEED initiative? So CDFT, as I showed you before, was one of the 6 training centers in the first phase. And they have trained about 14 faculty from government medical colleges who have now gone on to establish genetic diagnostic centers in their respective hospitals. And CDFT also took care of outreach centers in 2 aspirational districts in Yadgir and Raichur in Karnataka. So these are some of the photographs from the aspirational district that was being taken care of by CDFT. And they also came up with this brilliant idea of setting up a museum at RIMS Raichur. And in fact, you can see most of the things in the museum have been made by the students and staff of the diagnostics division of CDFD. So I think they really deserve applause for this initiative. And at NIMS, so NIMS Hyderabad was one of the 5 centers across India to be granted the NIMS Nidan Kendra project in the first phase. And through that, we have been able to establish a state-of-the-art cytogenetics and molecular genetics lab in NIMS. And in the second phase, NIMS has been designated as one of the training centers. So we have started our fellowship in genetic diagnostics, and that started in April 2025. So the first batch is now undergoing that training. So these are the facilities that we have been able to establish at NIMS through the Nidan Kendra project, and the Sanger sequencer and all the other equipment that is required for molecular genetic and cytogenetic testing has been procured, and a lot of tests have been standardized, and the diagnostic facilities are now smoothly underway. And these are the clinical facilities, counseling, and prenatal diagnostic facilities that we offer at our center. And because of this quality care and this high quality work that is going on in both the centers, together we were designated as a joint center of excellence for rare diseases under the National Policy for Rare Diseases in 2021. So in fact, one of the things of the UMID program is that the Nidan Kendras, which are established under UMID, are supposed to then participate in this NPRD and contribute to that and help in the diagnosis and management of patients with rare diseases. And as of now, there are 13 centers of excellence under NPRD. In the next talk, there'll be more details covered about the National Policy for Rare Diseases. And I'm happy to say that NIMS CDFD, the joint COE, is one of the 13 centers being recognized as of now. So what were the outcomes of this program? As of now, of course, it's an ongoing program. So if we do a midterm assessment, we can see that it has greatly helped in capacity building in the country for trained manpower who are trained in medical genetics, capacity building for genetic diagnostic facilities at medical colleges, and it has also demonstrated very well the feasibility of implementation of antenatal and newborn screening programs at the district level. So the whole idea of the program is to bridge this gap between the apex institutes and medical genetics centers where all the good work and research is going on, and ensure that it reaches the community, the grassroots, where it really makes a difference. So I'll stop with that. Thank you. And this is a small poem I had composed in Hindi, which includes both Umid and Nidan. So yeah, thank you. So I think we'll take the questions in the end.
So thank you very much, Dr. Pragna. And the fourth program, which is initiated by the Government of India, is the National Policy on Rare Diseases, which is called as NPRD, as mentioned by Dr. Pragna. This is initiated by the Ministry of Health and Family Welfare, and the main goal is to do diagnostics and research as well as management of patient with rare diseases. So now I request Dr. Shagun, who is a professor and HOD, and who is involved in the management of patients under NPRD, to just give more details about this project. So good afternoon, everyone. So we have already heard from the previous speakers about the wonderful initiatives by the Government of India and the Department of Biotechnology towards the establishment of advanced research facility, diagnostic facility in the field of genetic diagnosis. So now this is another wonderful initiative, and it's, I would say, a one of a kind policy in the world, which actually now helps in treatment of patients with rare diseases, majority of which are genetic diseases indeed. And this is something which really brings hope to many such families, because all these years, of course, there are challenges in diagnosis and all, but one of the major challenge which remained is the treatment of genetic diseases. So for many diseases, of course, even now the treatment is not really that much available, but for some diseases, at least the treatments have been available. Some of them in India, but majority of them again highly prohibitive by cost, or the drugs or the therapy not being available in India. And now this is a policy which actually kind of enables these therapies to reach the patients who and their families who are suffering from these conditions. And let's see how this has been framed and what are the various components of this. So to press the center. So, okay, slide moving. Moving actually pressed. So we've seen that as of now, we know that there are around 7,000 rare diseases. The number may be even larger, because still we know that practically almost every week, 1 or 2 rare diseases do get discovered. So of these 7,000 rare diseases, what are currently known, a huge majority are indeed because of a genetic etiology or a genetic cause. And as such, the what is a rare disease, that definition itself is really difficult. So different countries, even WHO has different definitions of what to call a rare disease. And in fact, for India, we don't really have a clear-cut definition also, because we don't have the complete epidemiology also for a rare disease for our country. We don't know what is how we would say how rare or how common for our country. But on an average, mostly we say that a rare disease is a disease which affects around 1 in 2,000 people. So over the world, across the world, around 350 million people are suffering from one or the other rare disease. And even in India, the burden is really huge. So around 70 to 100 million people in India. Again, this is an estimate. So we don't know the actual numbers. So this is the huge burden which we believe is of people who are affected with a rare disease. So there are the problems associated with the rare disease are, we could say, multifold. So there is a problem in diagnosing a rare disease, because most of the medical practitioners are not really aware of the symptoms and signs of a rare disease. So very often it takes a patient a visit to multiple doctors. So there are studies which say that on an average, a patient has to visit somewhere between 12 to 16 doctors before the diagnosis of their rare disease is established. And this, on an average, again, the duration is somewhere, they say, a mean of around 5 years. So again, majority of these diseases, of course, we see are there in are basically affect children. And for a huge majority, again, still treatment is not available. But for many now, treatment is is available now, and also for many, treatment has been available over the years, but not everything has been accessible. So the challenges for a rare disease patient are basically start right from the diagnosis. Again, because of the disease, a single as a whole, they may be a huge group, but a single rare disease would be something which not every doctor would be really familiar with. So making a diagnosis is a huge challenge. Treatment is a challenge. And the other big challenge is the prevention, especially for this group of diseases where we know a large majority don't really have a curative treatment as of date. So, and for a country like ours with our huge population, so prevention is the area which is actually very, very important. And here again, there is a challenge, because we don't really know what are the, across India, what are the various diseases prevalence in different communities, different ethnic groups, and how we could act in kind of identifying the carriers of these conditions and thereby offer them preventive strategies. So when this particular policy, when it was framed, it is kind of, the aim is to address all these aspects which are basically unique to this group of diseases. So this policy per se, it was launched in 2021. So the aims basically are on both the aspects. So one of the aim is to provide a kind of a comprehensive preventive strategy, because this is something which will go a long way in eventually reducing the burden of these diseases in the community. And the other immediate, in a way, something, the intervention which is important for the existing patient is to be able to provide them access to affordable health care. So these are the two main, we can say, the aims which are there for this policy. So what are the various provisions? So first, of course, under the policy, certain centers of excellences were identified. So initially there were 8 centers of excellence identified, out of which CDFD NIMS was a joint center, and then subsequently 4 more centers, and recently the 13th center has also been recognized as a center of excellence where these preventive, diagnostic, and therapeutic interventions will be provided for patients. Then, of course, another component is to create the health personnel capacity, because we know that, there is a problem with respect to the awareness of medical practitioners towards these diseases. So one of the objectives or one of the deliverables of the policy is that we should be able to train more and more doctors in the periphery. So again, the centers of excellence are supposed to be the hubs through which the various other medical colleges would be connected, and this and their staff and their faculty and residents would also be trained in how to suspect a rare disease patient, how to perform a genetic diagnosis, counseling, and how to be able to refer a patient as needed. And then the other important component is to provide infrastructure support. So for all the centers of excellence, there is a provision of funds to the tune of 5 crores, through which establishment of both diagnostic, preventive, various setups in terms of the lab setups or prenatal diagnostic setups. So all these are covered under the provisions of the policy. Then the most important thing, which is like really relevant for patients who are already suffering, is with respect to financial support for their treatment. So initially the support was committed to be 20 lakhs per patient. So every patient suffering from a rare disease was to be provided rupees 20 lakhs one time in for the treatment, which of course could be used as a one-time treatment or could be used as long as the patient required the treatment. And subsequently, it was realized that this amount will not be sufficient for most of the patients, and the amount was increased to 50 lakhs. So currently, every patient with a rare disease who's enrolled under this policy receives 50 lakh financial supports towards his or her treatment. And some other components which are still in process and are being developed is research towards indigenous manufacturing of the drugs. So many of the drugs which are needed for these rare disease patients have to be imported, are not really available in India. So, and the costs are really prohibitive. And for many such conditions where such drugs, so some of these are in fact very common conditions like Duchenne muscular dystrophy or spinal muscular atrophy, where the costs presently are really prohibitive. So there are thought process and discussions going on around indigenous development of such drugs in so that the cost can really be brought down. And of course, there is also plans to involve more medical colleges under a hub and spoke model, so that these all the diagnostic therapeutic services could be extended to the grassroots level. So of course, NPRD per se cannot work in silos. So it is supposed to kind of interact and have this support from the DBT Nidan Kendras with terms of providing the diagnostic services, as well as the ICMR Rare Disease Registry, which actually is collecting data with respect to prevalence of the rare diseases in the country, because unfortunately, as a country, we still don't have the data. So ICMR is working in parallel to on this rare disease registry, which is going on since the last more than 4 years now. So these three components of the government are basically the various schemes are working together to kind of being able to implement this policy in a more holistic manner. So now coming to the treatment aspect. So at present, the treatment is basically the rare diseases have been divided into 3 groups. So the first group are conditions which are amenable to a one-time curative intervention. So this includes conditions like various lysosomal storage disorders or various other conditions, whereby basically the bone marrow transplant or hematopoietic stem cell transplantation is the one-time curative treatment of choice. So this also includes conditions like various inborn errors of immunity or aplastic anemias or osteopetrosis, where this could be a one-time treatment, and this is something which is very much within the cost or the the cap which we have of 50 lakhs. The other aspect is other organ transplantations like liver transplantations for various metabolic liver diseases and certain other small molecule diseases, and renal transplantation for cystic kidney diseases and others. So this is these are group of condition where basically the CO is where transplant facilities are available. So under the policy, this patient would get a complete financial support towards the transplantation, and this would be a one-time curative treatment for this condition. And subsequently, we have the next group, which is the group 2. And these are diseases which do not have a one-time solution. So here the patient does need a lifelong treatment. And this treatment, however, has a relatively lower cost. So again, most of these patients are able to get treatment through that 50 lakh funding for a reasonable period of time, very often lifetime. So this again includes mainly two groups of further two subgroups. So one are conditions which are all these small molecule diseases like various aminoacidopathies or organic acidemias, where we have certain dietary formulae or certain special foods, basically supplements, whereby for that particular metabolic disorder, the specific diet can be given, and that helps in ameliorating the symptoms to a large extent. And then we have the other kind of conditions where again there are certain other drugs, maybe in the form of hormones or some other medicinal compounds, which are able to again help in the specific particular conditions. So some of them are listed here. So for tyrosinemia type 1, which is again a type of metabolic liver disease, we have a drug called nitisinone. So again, now it is now indigenously manufactured, and the cost has really come down as compared to the previous cost. Then we have osteogenesis imperfecta, which is a common condition of fragile bones. Then there we have various conditions where short stature is an important morbidity, and their growth hormone therapy is supported. And likewise, there are many other genetic conditions whereby specific medicines, we could say, are available, and these could be practically supported lifetime for that patient. But the most challenging group actually is the group 3. So this is a group where we have conditions for which some definitive treatment is available, but either the cost is really high, or it has to be, and it mostly has to be a lifelong therapy. So it's not a one-time solution also. So again, here there are two further subgroups. So there are some conditions where yes, okay, there is a lifelong therapy required, cost is high, but at least the definitive benefit is proven. So this includes various conditions where enzyme replacement therapy modality is there. So some of this like Gaucher disease, Pompe disease, and some others. So these are few conditions where there are very good therapies available. It's just that they have to be given lifelong, and their cost is really high. And then we have the group 3B, which actually includes two of the most common conditions which are there. That's Duchenne muscular dystrophy and spinal muscular atrophy, where we have, yes, treatments available, but the cost, of course, is very high. And also the definitive benefit right now, it's a bit preliminary to say whether those particular drugs which have now become available, how much long-term benefits are they really going to have for this group of condition. But yes, the preliminary data is showing at least some beneficial effects, especially for the condition called as spinal muscular atrophy, that's SMA. So, but this is the group actually where the challenge arises, because here basically the 50 lakh amount is not really sufficient for these patients. So either the drug is too high, or the cost is too high, or the duration itself makes it like the 50 lakh would hardly cover for an year or 1.5 for that particular patient. So which for a for a genetic rare disease where mostly a lifelong therapy is required, a 1 or 1.5 year therapy is hardly of any use. So this is actually so for many of these group 3 diseases, so because that 50 lakhs is not sufficient, so another avenue which the ministry has created is a digital portal for crowdfunding. So all the patients who get enrolled under this policy through a COE, their names, their photographs, their disease diagnosis are uploaded on this particular portal. And then there is a button which, on the portal through which anybody can make a donation in name of a particular patient and a particular center. So the donation goes only to that specific patient. So as of now, we can see some of the data here, that these are the total number of patients which are enrolled all across all the 13 centers. And then this, unfortunately, but the donation have not been really, I mean, we could say they are like hardly very minuscule and would hardly support even one patient's treatment. So these are some of the institutes, the Center of Excellences. So as I mentioned, 13 are there. And of course, CDFD with NIMS is one of the Center of Excellence under this policy. And if we go through this page, we can also get much more interesting information. So you can see some of the common conditions and also their treatment protocols, drugs available are mentioned on the website, which any of you can look up if you're interested. And then this is the page whereby we can see the individual patient's information, and you can, if interested, we can make a donation through this particular tab which is there. So now coming on to the implementation of this policy at our center. So as I mentioned, so we are a joint center, CDFD and NIMS. So in NIMS, of course, we have our clinical facility where we have 2 permanent faculty and 1 contractual faculty. And we have the DRNB medical genetics program. So we have the students and we have counselor students. And then, of course, we have the other supportive staff. And at CDFD, we know that we have all the research and the diagnostic staff and the state-of-the-art diagnostic facilities there. So this is the statistics for our department for the last year. So last year, we have seen around 12,000 plus patients with rare diseases, performed around close to 20,000 genetic tests, also performed preventive measures in the form of prenatal procedures, around 500, same 500 plus antenatal ultrasound, fetal autopsies, and also 4,000 plus genetic counseling sessions. And these, of course, are the laboratory facilities at the Nidan Kendra at NIMS, at the diagnostic lab at CDFT. And this is a patient volunteer from ORDI, the Organization for Rare Disease from India, Ms. Shravya. And these are the records which we are maintaining. These are the forms which are filled up for every patient who comes to our COE. So there are 2 sets of forms. One is for uploading into the crowdfunding portal, and the other set is for the listed conditions. So under the 3 groups, so basically we have 63 conditions which are listed and approved for treatment right now. So whoever patient belongs to those 63 conditions, they will be filling in a form which is specifically for financial support. So there are 2 sets of forms. And then, of course, these are just pictures of some of the medicines which have been procured for the patients. So at present, our center has uploaded around 527 applications for crowdfunding on the digital portal of the ministry, and 151 patients with rare diseases are presently receiving treatment at our center. Most of them belong to the group 2. And there are few patients, basically these are patients who are receiving enzyme replacement therapy from the group 3. So these are some of the conditions which are being treated at our center. The largest number is the Gaucher disease patients who are receiving enzyme replacement therapy. And then many others are basically small molecule diseases. There are also certain inborn errors of immunity patients who are receiving therapeutic support at our center. So last year, we had started our daycare facility. So this was launched in September 2024. Every month, we give around 40 to 50 ERT infusions. So as of now, we have given around 500 plus ERT infusions, all through financial support through the policy for 32 patients. So 27 of them have Gaucher disease and 5 of them have Pompe disease. And in addition, we also have prenatal diagnostic services. So we had started our dedicated fetal medicine clinic last year. Again, around 800 plus patients have undergone ultrasonography or prenatal invasive diagnostic procedures at the center. In addition, we conduct various awareness programs for other doctors also, and also some of the programs we have involved patient support groups also. So this was a Rare Disease Day in 2023, which we had celebrated. We had another CME last year on management of genetic disorders, another CME on thalassemia, where in fact the Thalassemia Society of Hyderabad, many of the children and their families participated in this. Then we conducted certain workshop for fetal medicine doctors for basically genetic testing and genetic counseling workshops. And subsequently, we had a sensitization workshop organized under the Ministry of Health and Family Welfare, when one of the representatives from the ministry also was participant. And this was mainly meant for, again, the various district medical college faculty. So faculty from every medical college of the district, we had one obstetrician, one pediatrician, and certain other colleges, we also had certain laboratory faculty who had attended this particular workshop. So actually the aim of this workshop was to sensitize and educate the other medical college faculty for creating awareness regarding rare diseases and how to help these patients with diagnosis and treatment support. And in addition, we also conducted a patient awareness activity, which was actually led by our genetic counselors. So this was done in March this year. And then our activities have been covered in major dailies, so including Times of India, Hindu, Deccan Chronicle, whereby our statistics and how we are helping in management as well as diagnosis research for rare disease patient was documented. So in addition, we have various research projects going on, many of them in collaboration with CDFD. And we have various training programs which do exist at NIMS. And I think that would be all. So thank you very much. Thank you, Dr. Shagun. Now, I request all the speakers to have their seat on the dais to have an interactive session. Arpit. Mics. First slide. Photos. Photo ले रहे हैं. Should be highlighted. Camera should be there. Yeah. So now I request Dr. Ashwin Dalal to just felicitate our speakers. Quick. Come this side. इधर take. Thank you, Dr. Dalal. So now, any questions are there? You can please you're welcome to ask questions, and even online. I could see one hand raised. Dr. Hari.
So I was always confused about this reference genome, right? So now that with this, so this variance that you have compared, the list, the numbers you have given, is it with reference to the so-called universal reference genome for the whole world, or is there something now after this, something will come up for India, and using that would be a better way to do things?
Yeah. So one thing is the variants that we mentioned is with respect to one specific reference genome version, which is predominantly used in diagnostics, right? So, but world is moving away from using one single reference genome. Now, there are approaches which incorporate all these different variations into something like a pan genome or a graph genome. But we are at present, if you look at any specific how people use this kind of information in a clinic, we still use that reference genome. Like HG38 is what most people use. So the data set that we have provided is against that reference genome, which means that anyone who has used the same reference can use the Genome India data as the baseline. As we move further, these will be reinterpreted so that it stays updated. So that would be my answer for now. But yeah, there's no single reference genome that we hope to make with Genome India or anything.
So just one more. I just thought, so now with that, let's say Prajad, and they are sequencing, and then you have your 10,000 genomes. Will the variants, for example, that shows up as a disease, if it is found in this 10,000, it means probably there are more complicated things going on that not always, I mean, those sort of comparisons, can it be done?
Yeah, of course. So that kind of analysis is only as good as the amount of metadata you have. So there would be, let's say, modifier mutations which decide how.
Right.
Like
But
what is-
comparing will help us to know
Yeah,
whether,
absolutely,
those sort of
absolutely.
things are
Yeah.
there. And just one last one for the rare disease thing. So you said 80% are of genetic origin, right? So out of that, how many we know a causal, I mean, a variant that's likely to cause, or very strong correlation between a variant, or is there a large majority for which, we don't know the genetic basis?
Yeah, so firstly, the question is how many genes we know diseases. So we have 19,500 genes roughly in our cells, and we know for about 7,000 to 7,500 genes we know disease they cause. So for about 13,000 genes, we still don't know what disease they cause. And now if we do the exome genome sequencing in about 100 patients, we'll get a definitive diagnostics roughly in about 30 to 40%. So that is what about 50% or more patients remain without a diagnosis even after doing all these tests. So we still don't know a lot, and that is why all these databases are important.
Thank you.
So any more questions? Yeah, there. Yeah, you can give Charvi.
Hello, Dr. Tej. It was a very nice talk. Actually, I was curious to know about there are few blue circle that was clustered within the Tibeto-Burman group. Could you please mention what kind of population it is?
So I can't specifically mention any population name, but the thing is, if you look at the Austroasiatic groups, they are spread. There, a lot of pockets are in AP, Odisha, that kind of region, but you also have some pockets going further northeast, right? So those would be the likely populations which would cluster there.
Okay, thank you.
Any more questions? Yeah, Nirja.
More of information seeking from the perspective of parents and other societal persons. Like if the listed disorders for which the NPRD policy is supporting the patients, how likely is the addition of new diseases coming under NPRD, and how much of this information is going to be extended to the general patients?
So actually inclusion of, I mean, there is a very much a provision for inclusion of additional diseases as long as, I mean, certain criteria are satisfied. So one, of course, that it should be a rare disease. Then preferably, there should be a genetic report saying that it's a rare disease of genetic basis. And then we should have a treatment which is available and which has been approved, maybe, by FDA or EMA. We should have sufficient literature saying that, preferably larger clinical trials or, larger data saying that that particular treatment is of proven benefit. So on basis of all these things, and of course, the cost factor, I mean, that it should at least be beneficial to the patient for that 50 lakhs, should be able to give a meaningful benefit to the patient. So on the basis of that, on a case to case basis, because there could be, a rare disease for which, for one center of excellence, we may just get a single patient coming in, right? And it may not be there in that list. So if that particular patient, that condition does satisfy most of these, I mean, the criteria, then we take that case, and this goes to the application actually, along with all the literature and the available, approvals, goes to another, specific committee at the level of the ministry, and then such, the case is discussed, and then if it is, I mean, considered to be, whatever fits the criteria to include under the policy, then it gets added. So you must have noticed, I mean, that initially the number of diseases approved were lesser, and then, the version, I mean, subsequently more diseases were added to that. So that's an ongoing process. And of course, I mean, it's open for, again, depends on a case to case. Patients could be taken up for that.
Thank you. And one question I have to Dr. Tej is, in the part, I mean, what is the likeliness of the continuation of Genome India project? Are there going to be a next phase, or if it is, there is a next phase, then is it going to be extended from the point of population pockets which you have selected, or will there be a separate space?
Okay, so whether there is going to be a phase 2 is beyond my pay grade. So, but I can tell you that there are deliberations, and it is not going to be just a population. I mean, now it will build upon this to sort of also address medical concerns, healthcare in general, and it's going to be that sort of a program. Yeah.
So according to you, should it be an extension of the areas which you have considered in your first phase, or what do you think it should be entirely a different spaced regions?
Yeah. So, like I said, it is there is definitely going to be a component of deliberate oversampling of underrepresented populations, for which we will still have to do targeted sampling. But it's not going to be just with the intention of forming a baseline anymore. That's what I was getting.
Thank you. Thanks a lot.
So, any other questions, Anjana?
I just wanted to ask, we have a list of patients with rare genetic disorders, as in Umeed, in Prajed, or in rare genetic policies also. So currently the treatment options may not be available, but in future, if they are available, can these patients be contacted back?
That we have an ICMR registry as well now, a rare diseases registry as well. Of course, right now that includes 6 specific categories of disorders, but that's also going to be expanded, and more conditions are going to be included in that. So all these patients' details are registered there. So as and when newer therapies come up, or some therapies become accessible and affordable, these patients would be contacted, called back, and would be provided access to those.
And what about clinical trials? Like, do they get
Yes.
enrolled into
So-
clinical trials also?
Yes. So one of the purposes of having this registry is also to identify patients who could be potentially enrolled into these clinical trials, as and when they are, planned and started.
So if somebody wants to develop a therapy for certain disorders, anyway, whatever is listed in that, so can these patient cells be taken, like, can we get those cells and then work on them? So that see in cell line, or in some other form, if the treatment is working or not, and then try to
So
develop
that
something
depends
out of
which
that.
condition we are talking about and which project or which program we are talking about. So the ICMR Rare Diseases Registry doesn't have any biobanking.
Okay.
It's just a registry, purely a registry as of now. Some of these other things like Project and all, we are doing biobanking
that
of
do,
samples.
we do.
So that with proper approvals and all could be accessed for further studies.
Okay, one more question.
Yeah, thank you, ma'am, for a very wonderful today's evening session. That you mentioned that NPRD Act, if you see earlier around, I think 2024 August, it is released. Okay, how frequently these, the editions, whatever madam is asked, that extension? That is first one. Can you?
Dr. Shiv.
So it's not frequently that the diseases are added. Diseases are added, as Dr. Shagun said, depending on what representation is given to the Central Technical Committee. If a clinician sends to the Central Technical Committee that there is a patient who has a report and this therapy has been approved by FDA or some European authority and the benefit has been proven, then the committee deliberates and that disease is included. So almost every 2-3 months one or two diseases are added. Sometimes it may take 6 months. It depends on who represents to the committee.
Yeah, correct, sir. But actually, if you see, I'm the representing the group 3B category of the diseases, the parent advocacy team, Bharat MD Foundation. If you see that Duchenne muscular dystrophy, there is a treatment which we have mentioned in our NPRD Act. But actually, maybe you are more knowledgeable than us that EMA and then FDA is also not, keeping that in their list. But still, we are keeping that in our, policy. So for that purpose, I ask you that how frequently we, review that, our recommendations or the information in that.
Yeah, so in policy they had classified these diseases, but then it was very clear that financially it is not going to be viable if we have to import these therapies and whether they are really effective or not. And that is why a lot of emphasis now is given by Government of India to develop these therapies in India, because reliance on importing these therapies is never going to be, the money is never going to be enough to cater to such a large population as ours. So we have to work in that direction. And also we have to see, because just because somebody is saying therapy is available, it does not mean that it is effective. When you want to use the government money or the taxpayer money, we have to be sure that it is really beneficial and not just claimed by one company that it is beneficial. So that is why the technical committee takes a review and then decides. The classification of diseases was from the point of view of how they could be taken up in case there is enough funding available. But it is very obvious that these medicines, if they are imported, they are going to cost such a lot that it is not feasible for government to handle this.
And also, sir, one more thing for the Nidhan Kendras, are we are restricting this one only to import this genetic knowledge to our doctors, or for some of the diseases, the procedures also and the protocols also defined? Do we have any plans for CMEs to educate or knowledge transfer for this protocol implementation for the patients?
Yeah, so we do patient awareness programs also. I think last year we've done one. So it's not just for doctors. So we do different types of programs. This year, so far, probably we've not done a patient awareness program. This year's programs are all related to, probably there was a thing in our department where the genetic counselors took the lead, and there was some information given about common genetic disorders and the counseling aspects and all. But on a larger scale, we did do a patient awareness program for patients with lysosomal storage disorders sometime last year. And so we do conduct both kinds of programs. It's not just restricted to medical professionals.
Because as with the pediatric that's project, we are able to get the samples from almost remote areas of the almost entire country. Now we are finding next step, like need even looking for how we can help those patients in management to provide the quality of life, so that if you can provide CMEs for the treating doctors also, it will help the community in a larger
So
extent.
that's the reason why we focus a lot on educating medical personnel, because finally they are the ones who are actually going to be going out to the primary healthcare level and actually treating patients. So this year we have, with the support of the Ministry of Health and Family Welfare, in February we had a program, an awareness program on rare diseases, where faculty doctors from government medical colleges from across Telangana participated, and we had a lot of deliberations on all these various aspects of diagnosis, management, prevention, prenatal testing, all of those.
So can we expect any one more program in near future?
Maybe I request you can
So just
interact
to-
during high
yeah.
tea.
So I just want to add, so in fact, we would be very happy. I've been previous program also, as Dr. Pragna mentioned, for lysosomal storage disorders. So, and also subsequently, we had a program for thalassemia patients. So actually, both of them involved, were strongly involved with support of a patient support group, right? So for the LSD, I mean, it was through the ORDI, whereas for the thalassemia program, it was with the Thalassemia Society of Hyderabad. So because of which many patients were already, part of that particular support group, and they are, emotionally invested, attached, and they are actually, having that, what do you say? I mean, a unity and kind of. So, I mean, I believe your foundation also, I mean, we are aware, we are following your work. So many muscular dystrophy patients are attached to your organization. So in fact, I would say this would be, it's a more of a teamwork in that sense, because we can reach the patients who are coming to us, but having them as a group where they can discuss, where they can raise questions about their management or their other supportive issues, and also that's why we conduct these kind of programs. Also, we have done it for LSDs, for thalassemia. We would be very happy to do it for muscular dystrophy patients, because besides educated medical professional, again, patients being aware and patients knowing what is the channel for them to go about in terms of approaching a particular or whatever a particular facility which is there, or a service which is there. I mean, it has to be, I think, a joint effort, or effort by multiple people coming together. So
So we have one question from online. Can you just use the mic?
Dr. Shubha Fatke ma'am has posted this question. Have some medicines developed in India under Make in India policy?
Yeah.
So, as of now, there are through the Niti Ayog's initiative, so 4 medicines have been developed in India. The predominant one being the nitisinone, which is used for treatment of tyrosinemia. So the nitisinone, which is used in tyrosinemia, that is being made by 2 companies already in India, and then there are 3 more medicines. One is for seizure disorder, and there are 2 more I'm not remembering, but 4 drugs. Eliglustat also is being manufactured in India. So as of now, there are 4, and hopefully more will start being. And triantine also, yeah. So triantine for the Wilson's disease, eliglustat, nitisinone, and the seizure disorder drug.
Okay. So with this,
Gupta, this is Ramchandar. Hello.
yeah, Dr. Ramchandar.
Yeah, actually, can we access this Genome India database for our comparison?
To ask Tej.
Yeah.
Tej.
Is
When will it be available to public?
Yeah, so like I said, it's the data is deposited at IBDC, and recently there was a call for proposals by DVT where you can propose your project and you can apply for access. So I believe they're actually screening those proposals, and soon the results will be out. And on top of that, we're working on a variant browser. So that should be public again. That's when it will be public is controlled by IBDC. So once it is fully ready, that people can use that to ask the frequencies of whichever variant you're interested in.
No, Tej, why this now projects are required? Right now we are doing some project, it is there. Now we want to compare those variants with that one. So is it available right now? This is the question.
Yeah, so like I said, from our side, it's been already deposited with IBDC. So from there,
So now it
DVT
is not
controls
in
that.
Tej's
So
hands.
yeah.
How to access it?
Tej's hand.
Again, beyond the pay
Okay.
grade.
So thank you, Dr. Ramchandar. So now I thank all the experts. Now, as you know, so now as a part of this, एक photo लेना. So I'll also. Okay. So as a part of the symposium, we have organized for the first time essay competition for school children. So the topic what we have given them was how the genetic revolution will shape the future of India. So this is for the students for class 8 to 10. And to our surprise, we got so many essays and almost like 86 essays we have received. And then out of which we have selected 3 prizes, but joint prizes because 6 prizes we are giving them. And for this, I announce the prizes now.
Would firstly like to I would like to I'm very grateful for the opportunity presented by this event because it gave me an opportunity to go and explore about the topic, my interest that I've been wanting to explore for quite a while now. So my essay was comprehensive of a lot of topics. Firstly, I took a case study from Djibouti, a country in East Africa, which tried to solve its malaria issue by genetically modified mosquitoes. If countries like this can foster innovative solutions, why can't we in India? So I started with how genetics has developed in India and in the world, in fact, from Mendelism to Rosalind Franklin's Photo 51, the Human Genome Project International. And after that, I wanted to discuss issues in India. Through human health, we already are using PCR amplifying techniques, I believe, and RT-PCR served a lot during the pandemic. But we do have an issue of tuberculosis. In fact, India has 26% burden of tuberculosis today, and it is a socio-economic issue for us because it is a disease of the poor, of the slum dwellers, of the labor workers. And the thing is, the bacteria has developed resistance to all the antimicrobials that we use for it. So that is why we've started developing tests, I believe, that since, for example, rifamping bacteria, it has antimicrobial tests for that. And if we have more investment in this sector, maybe we can solve this issue, because it's a socio-economic responsibility for us as a country. More than that, in agricultural fields, we have it. Firstly, I think we made self-sufficient because of the green revolution, and we started hybridization, genome editing, and we have bt cotton techniques now. So the main thing that I drew from this entire project was, we have lots of issues to solve. We have lots of things that we can take up, and we can put our innovative solutions. And I think the sector of genetics has a lot to offer in that. And I think Shubha ma'am, in the starting, she mentioned that we have lots of ethical dilemmas to go about. So this is one thing I noticed, that when it comes to ethical dilemmas, uh, when Herbert Spencer and Charles Darwin first brought out their evolution theories and survival of the fittest, it was used by propaganda artists to actually justify their racial actions. For example, personalities like Hitler had justify their racial actions by using survival of the fittest theories. So that is why I felt that it is important for us to establish who engages with our genetic technologies. So I would really like to thank the organizers of this competition for providing me the opportunity. Thank you very much.
Congratulations, Apurva. I read your essay and it was beautifully written. And maybe we'll see you another 10 years down the line in CDFD or CCMD working here. All the best for your future.
And the joint second prize is to Vaisaki Mulugu from Santa Maria. Thank you, Dr. Pragna. So now I invite Tej to give our first prize. The first prize go to Adya Agarwal from Santa Maria. Sana. Yeah. Which class you're? She's 10. Take the mic and yeah. Yeah.
Hello, everyone. Thank you for this opportunity in this competition. My essay mainly focused on how it can be used to fight hunger and diseases, and how it can really help in economic growth and development of India, and saving countless lives, and help poor people achieve the medical resources they need in genetics diseases. Thank you.
Yeah. Thank you. Thank you, Tej. And I should thank her because she's coming from the other part of the city beyond where I come from. So with this, what is the program? Yes. I can announce the joint winner of the first prize. This is Ishanvi Agarwal. She's also from Santa Maria, but she could not come here. We congratulate. And I congratulate all the students for this winning the prize. So now I request all of you to join us for it, high tea. Yeah, and a group photograph with the children. You can come to the dais. Group photograph. Come.
Come, all of
you can join. Yes. Gauri, Gauri, Aishwarya, Charlie.
आगे पीछे जाओ
కాదు ఇది ఎందుకు మధ్యలో ఆన్లైన్ మ్యూట్ అయిపోతుంది అన్మ్యూట్ అవుతుంది అవ్వింది we have got and we have done this సో కాదు మ్యూట్ అయింది వాళ్ళు ఫోన్ చేశారు అందుకనే మేము ఇది చేసాము ఇక్కడ అయింది కదా సార్ వచ్చి అన్మ్యూట్ చేశారు సార్ ఇప్పుడు చేశారు మీరు చేయలేదంటారేంటి ఇప్పుడు
హలో
హైట్ కాదు అన్మ్యూట్ పవన్ పైన థర్డ్ ఫ్లోర్ కి రా హైట్ ఉంది కొంచెం తాగదు స్నాక్స్ ఉంది తొందరగా మ్యూట్ గా
online participants, we thank you for your attendance. We are leaving the session right now. Thank you. Please log off. And ఇంకా ఉన్నారు. People are there.
People are there.
So you leave the meeting.
I'm not the host. They only have to be CDFD.
CDFD. This is
అదే